Good morning. It's August 2nd, and this is your daily brief in science. Here's everything you need to know. A new study from Queen Mary University of London sheds light on the complexities of photosynthesis in varying stellar environments. This collaborative research focuses on the potential for oxygenic photosynthesis on exoplanets that orbit low-mass M-dwarf stars, which are common in our galaxy. By investigating how these stars' light affects photosynthetic performance, the study contributes valuable insights into extraterrestrial light harvesting systems. The findings reveal that plant-like antennas face challenges in adapting to light from stars with temperatures below 3,400 Kelvin. This limitation suggests that simply increasing the size of these antennas may not be a viable solution. The researchers also examined how the spectral fluxes from these lower-mass stars impact photosynthetic efficiency, proposing enhancements through the addition of more light-harvesting complex 2 trimers. To further optimize light capture, the study introduces a complementary lattice diffusion model. This model treats excitations as localized entities that hop between clusters of pigments, potentially improving light harvesting capabilities. The researchers argue that while current antenna structures can adapt to low light, there is a need for further evolution and optimization to overcome entropic barriers. Moreover, introducing an energetic gradient to the antenna significantly boosts efficiency and electron output. Plants naturally regulate photon absorption and can downregulate their antenna size to prevent photoinhibition in low-light conditions. The architecture of the antenna reaction center plays a crucial role in maximizing light capture, suggesting that similar adaptations may be necessary for organisms on exoplanets. The authors, including Thomas J. Haworth and Edward Gillen, received support from various research councils, and no conflicts of interest were reported, indicating an objective approach to their findings. Eli Lilly has unveiled promising results from a late-stage clinical trial of tirzepatide, known as Mounjaro for diabetes, and Zepbound for weight loss. This drug has shown a significant ability to reduce heart failure risks in patients with obesity. The trial included 731 participants who were suffering from obesity and heart failure with preserved ejection fraction, or HFpEF. Results indicated that those taking tirzepatide experienced a 38% lower risk of hospitalization, increased heart failure medication, or death compared to the placebo group. In addition to these heart failure benefits, patients taking Zepbound reported an average body weight reduction of 15.7%. After 1 year, participants also demonstrated significant improvements in symptoms and quality of life, as measured by the Kansas City Cardiomyopathy Questionnaire. Dr. Patricia Pellica from Mayo Clinic praised these findings, calling them a significant advancement in treatment options for heart failure patients and highlighting the drug's potential to enhance quality of life. Eli Lilly plans to submit the trial results to the U.S. Food and Drug Administration later this year, which could lead to Zepbound being included in Medicare coverage for obesity-related heart failure. Following this announcement, Eli Lilly's shares rose nearly 3%, reflecting a more than 40% increase in value for the year, indicating strong investor confidence in the drug's future. Additionally, the company announced that the shortage of Zepbound is expected to end soon, which may affect the market for off-brand or compounded versions that emerged during the supply constraints. This trial positions Zepbound as the second GLP-1 drug to demonstrate efficacy in treating heart failure, following Novo Nordisk's Wegovy. With heart failure affecting approximately 6.7 million adults in the U.S. and accounting for nearly 14% of deaths in 2022, the findings from this trial hold significant relevance. A recent study published in the journal Science has unveiled intriguing connections between plant prickles and an ancient gene family known as lonely guy, or LOG. This research highlights that the prickles found on roses and certain crops are distinctly different from thorns, which are modified structures. Scientists have pinpointed the specific gene responsible for the presence of these prickles, revealing that their evolution stems from a single gene, showcasing the complexity of biological processes involved. Conducted by an international team, the study sheds light on the evolutionary history of prickly plants, which have been around for at least 400 million years. This discovery paves the way for potential genetic modifications in agricultural and ornamental crops, with the aim of developing prickle-free varieties. Notably, research led by Satterley indicated that mutations in the LOG genes could lead to the absence of prickles in eggplants, suggesting broader applications of this genetic insight. The findings also indicate that the formation and loss of prickles may be linked to LOG-related genes, exemplifying convergent evolution across various plant species. Historical practices show that early farmers likely bred out prickles from wild plants to enhance their ease of cultivation and consumption, which has significant implications for modern agriculture. Removing prickles could simplify harvesting processes and improve the marketability of certain fruits, like desert raisins, which face challenges due to their prickly nature. Zachary Lipman, a plant geneticist, has studied prickles in the context of crops such as potatoes, tomatoes, and eggplants, which lack these features. It's worth noting that while many believe roses have thorns, they actually possess prickles, which are epidermal outgrowths and not modified organs. Understanding the mechanisms behind the evolution of these traits could illuminate broader questions in evolutionary biology and deepen our comprehension of plant life. A significant discovery has emerged from southern China, where researchers have unearthed fossils of a marine creature named Schischania aculeata, dating back approximately 514 million years. This finding provides new insights into the early evolution of mollusks, a group that includes snails, clams, and octopuses. The fossils were discovered by doctoral student Guangshu Zhang during a road construction project in Yunnan province. Initially, these fossils were misidentified due to their unusual appearance, but Zhang made a pivotal comparison to a plastic bag, leading to the recognition of their significance. The fossils reveal a flat, slug-like body covered in hollow, cone-shaped spines made of chitin. These well-preserved specimens showcase soft body parts, including a muscular foot, suggesting that Schischania likely moved along the seafloor in a manner similar to modern slugs. Dr. Luke Perry from the University of Oxford noted that the spiny exterior of Schischania predates the development of shells in mollusks, enhancing our understanding of their evolutionary history. Published in the journal Science, this research highlights a very early stage in molluscan evolution, showcasing primitive characteristics of these ancient creatures. The discovery adds vital information regarding the ecological adaptations of early mollusks during the Cambrian explosion, indicating that Schischania likely fed on algae and organic matter in shallow marine environments. Moreover, the research suggests that the spiny structures seen in modern mollusks evolved from similar organic forms found in Schischania, indicating a shared evolutionary pathway. Experts believe this discovery could clarify the evolutionary timeline of mollusks, which, despite their diverse forms, have limited early fossil records. Peregrine falcons are soaring back in Yosemite National Park, thanks to a unique partnership between rock climbers and conservationists. Once on the brink of extinction, these majestic birds were declared federally endangered in 1970 and were added to California's endangered species list in 1971 due to drastic population declines linked to pesticide use. The widespread use of DDT after World War II resulted in thinner eggshells, leading to a catastrophic drop in peregrine falcon numbers, which dwindled to just 324 pairs in the U.S. by 1974. However, the ban on harmful pesticides in the early 1970s set the stage for their recovery, allowing the population to rebound to an estimated 40,000 individuals in North America by the late 1990s. A pivotal moment came in 1978 when climbers discovered an active peregrine nest on El Capitan. This discovery initiated significant conservation efforts, including the incubation of eggs in laboratories. Following a comprehensive 15-year recovery plan, the number of breeding pairs in Yosemite has risen from 8 in 2009 to 17 pairs in 2024. Since 2009, 51 nesting sites have been documented in Yosemite, resulting in the successful hatching of 385 baby peregrine falcons. Rock climbers have played a crucial role in this success story, actively reporting nests and assisting in egg incubation, which has allowed over 1,000 young peregrines to be returned to the wild. The peregrine falcon, which was completely absent from Yosemite after 1941, is now designated as a fully protected species in California. To protect these nesting sites, The National Park Service has implemented climbing route closures affecting less than 5% of the park's climbing routes, ensuring that climbers can continue their activities largely unaffected. Ken Yeager, a climber involved in the initial rescue efforts, expressed pride in the successful conservation of peregrine falcons, noting that without these collaborative efforts, the birds might have vanished from Yosemite. The National Park Service has reported a remarkable comeback of peregrine falcons since the 1970s, when there were no nesting pairs in Yosemite. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.